All right, it is my great pleasure to introduce Dr. David Zald, who currently serves as a Henry Rutgers Professor of Psychiatry and Director of the Center for Advanced Human Brain Imaging Research at Rutgers University. Before joining Rutgers, Dr. Zald spent 2 decades at Vanderbilt University, which is where most of us associate Dr. Zald with. Dr. Zald is internationally recognized for his work in affective neuroscience and neuroimaging. His research explores the neural substrates of emotion, motivation, and psychopathology. Using advanced MRI and PET imaging techniques, his work has illuminated how individual differences in these systems shape personality, decision-making, and vulnerability to mental health disorders. As a postdoc, I was fortunate to meet and work with him while working on projects examining how age-related changes in dopamine influence behavior. Dr. Zald's contributions have earned him fellowships in the Association for Psychological Sciences and the American Association for the Advancement of Science, and his work continues to shape the field of cognitive and affective neuroscience. Please join me in welcoming Dr. David Zald.
Thank you, Kendra. So I'm going to talk about a subject that someone the other day actually said, I didn't even know you were interested in this topic. And this really comes about out of some discussions that with Warren Taylor, who is a geriatric psychiatrist at Vanderbilt. And we had been discussing some things, and there are a number of threads of work from my lab that ended up sort of getting channeled into this discussion. And some of them were related to a long program of research where we were looking at measuring dopamine primarily with PET imaging, measuring different aspects of dopamine functioning and relating them to individual differences. There's a related line of work that was particularly about the role of dopamine and motivation and tying that into anhedonia. And a third line of work, which really came about somewhat by opportunistically over time, which was related to using some of the techniques that we were using for measuring dopamine functioning and applying that within the realm of aging research. Some of what I'm going to talk about came out in an article that Warren Taylor led a few years ago, though the way I'm going to present is a little bit more of my own spin on this, even though the essence of a number of the pieces of background for this are in this molecular psychiatry paper. Because this is going to weave together some different themes, I'm going to break this up into sort of several sections. The first, which is just a little bit specifically about late life depression. Then I'm going to talk a little bit about dopamine contributions to both motivation and psychomotor vigor. Then I'll get into a little bit of our literature on aging and dopamine, and then try to tie this together of how this is relevant in late life depression. In some ways, I see this talk as perhaps the opposite of vital longevity, because it's in some ways about a sort of non-vital longevity element, but hopefully in addressing that, you can lead to more vital longevity. So first off, a few comments about late life depression. The rates of major depression do not actually increase in later life. Depending upon the study and the way in the groups that you look at, there's some evidence that it may even decline somewhat. However, if you start looking at it in the context of individuals who have substantial medical problems, if you look at it in those who are living in long-term facilities and those who have developed disabilities, then you start to see substantial increases in rates of depression. Now, in some ways, that might seem unsurprising because those sorts of conditions are major health problems or disability. And in the general population at any age, you're going to see substantial increases in depression once you start to have those problems. So is there anything specific or different when we start talking about late life depression? Well, the first clue that there could be something different is if you look in mood disorder clinics for geriatric depression, you suddenly see lots of first-time episodes of depression occurring after age 50. And if you think about that, you say, look, these people were relatively invulnerable to depression up until now. They seem to have been relatively protected against what's a fairly common disorder to have, and yet now they're developing it. Could that just be because they've got other health problems coming up at that point? There's the first times that there's happening. Certainly that could be the case for some of them. But then you also start saying, are there any differences in the way it's expressed? And what you do see is that different symptoms show up at far greater frequency in older populations than they do in younger populations. And these include, for instance, greater levels of anhedonia, significantly more psychomotor retardation, more pronounced disability from the depression, greater cognitive problems often also associated with it. And then you see some other things tweaking in there, such as greater lack of insight, as well as, in some cases, an increased suspiciousness, sometimes going all the way into paranoid sort of thinking. At the same time, what you see is less prominent depressive ideation, and the vegetative symptoms actually can be somewhat mild, and remarkably also less of an anxiety component in the symptomatology. So you've moved towards what looks like a greater loss in the positive affective domain, while actually a lessening in some of the negative mood domain connected to it, or the negative affect domain. If we get, a little bit more specific into the anhedonia component, just in terms of to give you a sense of, well, how much more? Is this a little or is this a lot? You get almost twice as much endorsement of people saying that they enjoyed life less. In another study by Habels, where they did the diagnostic interview schedule, one of the variables that was most distinguishing was less interest in sex, where, that's a substantial component. Even in the younger, over half the subjects say that, but now we're up over 70% in the older subjects. So there is a change here in the distribution of symptoms, or the frequency of symptoms that are coming up. Importantly, those with the anhedonic symptoms, much more likely to show both disability. Also, they show increased mortality rates compared to those who do not have it. In this particular study, they essentially are comparing those with more dysphoric symptoms with and without anhedonia. And the presence of the anhedonia seems to have a substantial increase in driving both the disability and mortality. Going back to the psychomotor retardation, Alexopoulos, who's done much great work in the area of late life depression, estimates that the numbers are about 2 to 1 in terms of having a greater psychomotor slowing in late life depression compared to depressed individuals at a younger age. O'Brien, though their numbers are in frequency, the way they calculate it, are much lower. Similarly, though, a substantially significant greater amount of psychomotor retardation in the older life depression or late life depression. In terms of the cognitive deficits, about 30% of late life depression cases show abnormal performance on neuropsychological testing. This shows up on a number of different measures, ranging from things like response inhibition to verbal fluency, to working memory, cognitive flexibility. Alexa Poulos actually has referred to this as a depression executive dysfunction syndrome, really emphasizing the importance of these cognitive problems. And of course, there's been in the past conversations. I remember when I was doing more neuropsychological work, one of the first things we learned about in a dementia clinic was no dementia, depressed patients, with the idea that you could, if you helped with the depression, you could actually relieve some of those cognitive symptoms, even if those individuals were in fact more vulnerable down the road to develop dementia. You could at least temporarily relieve them if you could address the depression. In terms of that note, I'll recognize an error that should say LLD, not LDD. Sloppy on my part. Processing speed is really critical to these deficits. Yvette Shaleen, in a paper about 20 years ago, did some structural equation modeling of neuropsychological deficits in late life depression and found processing speed to be an absolutely essential core component that contributed to a wide range of performance deficits in late life depression. So just in summary regarding late life depression, here we're looking at the fact that we often see this as first emerging later in life. It's prominent, particularly prominent in those with depression who are showing disability with it, and that there's symptoms, some specific symptom domains that get particularly prominent, including anhedonia and psychomotor retardation. I'll note just as an aside here, it's also often inadequately treated. It's been estimated, and these papers are maybe a little, some of these papers are a little bit old at this point, but estimates are common to suggest that like 40% of cases with late life depression are either not treated at all or are very inadequately treated. For instance, they might be given some SSRIs, they're at a low dose, they don't necessarily work, and no one goes after it more aggressively. So the problem's not only there, it often doesn't seem to get treated all that. Switch gears a little bit here, and this is going to pull back not to something specific to late life depression, but more about processes related to motivation and psychomotor vigor and how dopamine may be related to these. And I want to just for a second, though, talk about anhedonia, because I've thrown the term out there, did nothing to define what was meant by that. The original definition of anhedonia was an inability to experience pleasure. And oftentimes, for instance, particularly in the schizophrenia literature, people will still often use that term specifically within the domain of an inability to experience pleasure. However, in general practice, it actually got expanded over the years. And so, for instance, in the DSM, they refer to markedly diminished interest or pleasure. Note that addition of interest. And that's really critical because there's a distinction now that we can make between just how you respond when you receive a reward versus your interest in those rewards, how engaged or motivated you might be or in anticipating those rewards that you can get. Now, those are obviously correlated. And when we, for instance, in our studies of depression, we routinely actually separately ask about how much someone desired things, how interested they were, and how they responded to those. And they're often correlated. There's no doubt that they're correlated. But should we actually be conflating them? There's good reason to think that we shouldn't. A good example of this going quite a ways back was an observation that Donald Klein made, which is that depressive patients often actually enjoyed rewards that were made available to them. They said they wouldn't be interested. They said they wouldn't, go out and do it. But when given those rewards, it's like mood brightening, like when something good happens, depressed patients actually will often show greater mood brightening than non-patients will. So it's not necessarily truly an inability to experience pleasure when they actually are exposed to it. If you actually look at correlated measures, you run correlations between people's reported desire and the reward response they have when exposed. The correlations are not that strong, in fact. And third, there's differential predictive value. Those who show low drive are far more likely to actually meet criteria for depression than those who say they have a loss of pleasure. Both of them are predictive of depression, but the lack of the motivation or drive to seek the rewards is actually far more predictive. It's also got some major prognostic value when you have it in depression. There are a number of different studies that I could be referencing here. I'm going to reference this one by Uher et al., which was from the STAR-D trials, as well as the GenDep clinical trials. These were both SSRI treatment trials, but those who had anhedonia far less likely to improve when given SSRIs than those who did not have anhedonia. Not only for medication treatment, you also see this with cognitive behavioral therapy. Michelle Krask has particularly published in this domain that traditional CBT is far less likely to be effective when you have individuals who show anhedonia. So it's a variable we actually want to pay a lot of attention to. So when Michael Treadway was a graduate student in my lab was working on a lot of questions related to dopamine, and we were reading work, particularly by people like Ken Berridge, who makes a distinction between wanting and liking. And Michael came in and said, I'm really curious about this paradigm that John Salamone uses in looking at dopamine functions and motivation. And the way this paradigm works is he put rodents into a tea maze. And in one direction, they could get a small amount of food. They didn't have to work at all for it. In another, they had to essentially climb over a barrier, but they would get better food for that. And in the normal situation, in the normal situation, so you got the rat, he's got the choice. Overwhelmingly, a normal rat has not had any depletion of their dopamine system. They'll overwhelmingly choose the harder effort option to get that higher reward. However, if you start blocking their D2 receptors, like you give them Haldol, they will be much less likely to go for the harder reward. They will come in here and they will just select the easier option. Now, there are a number of things that could be going on here, you'd say. For instance, one of the possibilities, maybe they're not as hungry. Maybe they don't, they're not experiencing the reward as well. But the dopamine depletion did not actually change their food selection or their consumption of the amount of food if freely available. It also didn't change any sort of evidence of hedonic responses, their actual facial responses when given a reward. What it stopped was their willingness to work for the reward or willingness to work for the larger reward. That, of course, feeds right into the Kent Berridge sort of model of that there's a distinction between wanting and liking, where Berridge will argue that wanting is closely tied to dopamine, where his model would argue that liking has other domain neural features. It is not specifically a dopaminergic dependent process. So we were interested in this paradigm, but we initially had a question of, well, how are we going to implement this? I mean, you could imagine us building in the hallways of the psychology department a bunch of barriers that people had to climb over. It seemed maybe today with virtual reality, we could figure out a way to do it. I certainly couldn't figure out a way without making people in the department really angry, like closing down the stairs every time we wanted to run an experiment. Luckily, we could run this alternatively with paradigms that others had used in rodents, where they did this based on choices of numbers of lever presses. And you could just have different lever presses where you do it like a small animal had just one press to get a small amount of food, or a lot of presses to get a better food reward. So we developed this task called the effort that we felt we could do, effort standing for effort expenditure for reward task. Michael was much better in thinking of acronyms than I ever would be. And essentially the way each trial would work is they would be given a choice where they're given information about how much money they could make for a high effort trial or a low effort choice. And the way we did this, which is not perfect, but works, is really easy to demonstrate, is if you just want to take your dominant hand for a second and just start doing a few finger presses with your index finger on your dominant hand. No one should find that very difficult. Okay, now what I want you to do is switch hands with your non-dominant hand pinky. I want you to make finger presses. Now, you can all do it, but unless you are a musician who has a very specific reason to have a really strong non-dominant pinky, I suspect all of you felt like, wow, that's more effortful to do it. It's not impossible. Otherwise, it'd be a lot harder to type on all your messages on your keyboard, but it's much more difficult. And that's essentially the choice we were making them do. A bunch of finger presses with their non-dominant pinky versus index finger. The other information that they got on each trial was the probability of reward, which turns out to, I always thought was just going to be a complicating matter, but it sometimes proves quite informative in helping figure out what's going on. So if we do this, we found that we could easily manipulate this with dopaminergic manipulations. The first dopaminergic manipulation we did was with amphetamine. So amphetamine causes the dopamine transporter to start spitting out dopamine instead of pulling it back in, increasing the amount of dopamine in the synapse. What you're looking at here is these are the low probability trials, a medium probability trials, and high probability trials. When we were at the high probability trials, amphetamine didn't make that much difference. Everyone's fairly close to ceiling. People are very responsive to the probability. They really don't want to spend a whole lot of time expending a lot of energy and effort if they don't think they're going to get rewarded. They're much more likely if they're going to get rewarded. But once we did give amphetamine at lower trials, it did enhance people's willingness to expend effort. In some ways, it's interesting that they're willing to expend effort here because it's not actually all that sensible. And you might wonder what that might mean in cases like mania, for instance, where people might expend a lot of effort on very high-risk sorts of situations that may not work out for them. Importantly, this looks very similar, though, to the rodent paradigms, where amphetamine also causes that increase in willingness to give effort. I'll note the rodent paradigms never play this game with probability as well. That would be a more complicated training, I think. I've never seen them do that. But this gave us really a sense that, okay, we're able to manipulate this and manipulate it specifically with a dopaminergic manipulation. Other evidence from animal studies also showed clear relations of these sorts of paradigms with dopaminergic functioning. So I've mentioned already haloperidol. You can mess with this with dopamine synthesis. You can also go at D1 receptors. So the Haldol is affecting D2 receptors. Here's a study where you can directly manipulate this by manipulating and blocking D1 receptors. So what does this sort of task look like if we, you can see this actually, I should say you can manipulate it, or these days, as in this study, genetically knocking out D1 receptors will have this a strong effect itself. So what does this look like in depression? Depressed subjects are less willing to expend effort to get a reward in this task. We see the difference at all reward levels. It becomes magnified. The higher the reward level is, the bigger difference. In both cases, they are responding to information here. Let me be clear. They're not just saying, no, I won't do anything. They do respond, but across the board, a less willingness to expend that effort. We can model this computationally, fairly simply, where you've got essentially the magnitude of the reward. You have essentially a discounting factor for effort. You have, I'm sorry, the effort is essentially discounting the reward value. The P here actually is signifies at what point the person starts to discount. And sort of like you can imagine, if it's not that much effort, it's not going to have an effect. It's only once the effort becomes enough that you start actually seeing an effect. If we look at this sort of modeling across patients with depression, one of the things that's striking, so in these are controls who are we're calculating essentially subjective value based on their choices. What we end up with, the controls overall are showing much more subjective value because they're showing less effort discounting. There's also a difference at what point they start effort discounting. Here, there are many of our depressed subjects who start discounting almost right off the bat, whereas we rarely see that in our healthy controls. And so this sort of threshold at which it starts even impacting, and then as that effort level increases, this just becomes more and more magnified. This sort of finding within depression on the effort, at least it has taken just as a sort of simple willingness to expend effort, been replicated now by multiple groups in different depression samples. To be clear, it's not universal. Not all patients with depression show this deficit. There is some evidence that it may actually be prognostic. In one case, Isabel Berrien, she was not using the effort task, but a different effort sort of paradigm. But she found that those who showed the greatest effort discounting were actually the most likely to relapse after they were withdrawn from their antidepressant medication, which is interesting. That's still one of the only studies I know of where they actually took this task and used it prognostically, but I found it quite intriguing. Can you manipulate this in someone who's depressed by manipulating dopamine? There's really only one study, and it's that I want to mention here. It's a very specific group here because all of these individuals who had depression were also individuals who showed high inflammatory markers, tied to a hypothesis that neuroinflammation is having an effect within the dopamine network that may particularly bias people with depression. So what these folks did was they were giving these individuals L-DOPA. L-DOPA is the classic precursor, which we use to treat Parkinson's. But so they're increasing basically the people's dopamine synthesis capacity. This is not a huge study, but strikingly, what they found was that giving L-DOPA to patients with depression who had these markers increase their willingness to perform effort in the effort task. It was, they saw this across different dosage levels. They particularly saw it at 150 milligram dose. Didn't seem to quite reach significant at the higher doses. I will say I have no idea why that dose is the sweet spot. I couldn't tell you why that is, but it is consistent with this idea that you can manipulate effort willingness in depression itself by manipulating dopamine. Now, that initially will raise a question, well, is there actually a dopamine synthesis deficit in depression in general? And the answer to that is, at least in general, no, there is not. There are not a whole lot of studies that have actually looked at dopamine synthesis capacity, which you would measure using F-DOPA PET. I'll note, if you look here, there's one of these three studies that's showing it, which is this study here, and which by Martineau, and I want to focus in on that for a second, because it's actually quite intriguing. Because they broke their subjects up into, here they've got just their comparison subjects, so these subjects don't have depression. These are depressed subjects in red who they described as having impulsivity. And then in blue are subjects who were depressed but had psychomotor retardation. And there in the left caudate, these individuals showed a decline in their F-DOPA, their dopamine synthesis capacity. Why this is specific to the caudate, you could imagine in terms of its role in more cognitive functions that this may be an issue. Why it's lateralized to the left, not necessarily sure. It's not a huge study. On the other hand, it's very intriguing that that's the place where you saw this synthesis deficit. Now, one thing I haven't talked about in terms of this psychomotor retardation is another reward element, which is vigor. And people like Yale Niv have created this really intriguing hypothesis that tonic dopamine levels influence vigor, in this case, the strength or rate of responding, particularly the change in responding that happens when things are rewarding. And interestingly, in a study, this is actually with healthy individuals, you can manipulate this vigor by increasing dopamine synthesis with L-DOPA. I'll note the way they display this makes it look like you're going in the opposite direction, but this is in fact the directions of someone who has higher vigor when given L-DOPA. Sticking though back with psychomotor slowing, the place where you talk the most about psychomotor slowing and dopamine is in the context of Parkinson's disease. We all know Parkinson's being caused by the degeneration of dopamine neurons, particularly in the substantia nigra. And within that Parkinson's literature, there's long been an attention to the fact that you can both have a motor slowing or bradykinesia, but you can also in the cognitive sphere have a cognitive slowing, often referred to as bradyphrenia. And that's usually discussed with the idea that the more lateral, dorsolateral part of the striatum and the putamen has more motor involvement, and that as you move more medially and get into caudate involvement, that's when you start to see more of the bradyphrenia. There's in fact significant correlations between that sort of slowing in Parkinson's and neuroimaging markers for the dopamine system, particularly using the dopamine transporter, which, since the dopamine transporter sits on the presynaptic side, can be used both as a measure index of dopamine transporters specifically, but also presynaptic integrity of the dopamine system. And there's a strong correlation between speed performance in measures and that dopamine transporter level in Parkinson's. So we've got really strong levels of evidence here in terms of both our connections between dopamine and willingness to expend effort, but also the speed of processing and vigor sort of domain. Okay, third part of this talk, which is actually a relatively brief part, but ties in here with why we started looking at dopamine and aging, and to begin with. So when I got to Vanderbilt, one of the things that was appealing was there was a neuroradiologist there, Kessler, who was working with falipride. And the beauty of falipride as a PET ligand, it's a high affinity D2 radioligand that let us look not only at dopamine in the striatum, but also look at D2 levels in the cortex as well. And I was working with this. I was doing some other raclopride studies at Montreal Neurological Institute. All of this was with young adults. In fact, most of our subjects were between 21 and 30. And Greg Samonez Larkin approached me as he was finishing up a postdoc at Stanford and said, I'm really interested in dopamine and aging and reward. Can I come do a postdoc with you? I was like, I'm not really an aging researcher, but I was aware of the fact that dopaminergic measures change substantially with age. And he seemed like a bright guy, and he turned out to be a phenomenal postdoc. And my connection through Kendra really comes through Greg. And so we started to do a bunch of studies looking at age effects on different dopaminergic variables. And one of the things that's just incredibly striking is how robust these age effects are. I mean, oftentimes I run a study and I'm trying to pull out these small percent changes, and every group difference that I've ever looked at with PET is overwhelmingly small compared to the age effects that we often get. It's strikingly, you get regional variation. Some areas are much faster than others in their decline. The D2 receptors are robustly affected, but you see them actually much greater, for instance, in cortical areas where the decline over age is just substantial, to the extent that I will bet money down. I mean, I'll take out my wallet, and if we went and imaged every one of you, I will bet that if you take Nick and me here and we compare to some of the, graduate students in the room or postdocs in the room, you graduate students or postdocs are going to kick Nick and my in terms of level. It's inevitable. It's not limited to D2. Here's from a meta-analysis. These are the across studies of correlations essentially between D1 receptor levels and age. If we look at this in terms of dopamine transporters, the effect again is really strong. The one place where we get a break from this somewhat is if we start looking at dopamine synthesis capacity, which is interesting because I've emphasized dopamine synthesis capacity here. If we look at it in the striatum, the net result, it's sort of mixed across studies, and some of this is methodological, but the net result is much smaller. It's still actually significant if we look at this at the level of cortex, but it's not nearly as strong. And if we actually do a comparison across measures here, we've got D1 here with a correlation with age of like minus 0.77 with, if we look at D2, we've got here for average here of minus 0.56 with DAT, minus 0.68. Whereas for synthesis capacity, you're down here on average at about minus 0.06 with the midbrain and particularly frontal cortex being more substantial, but the striatum being actually quite weak, which is where you have the most measurement, in fact, of the highest levels of dopamine synthesis showing up. A huge question, why is that preserved? Because if we actually look at postmortem studies, it's say like 4 to 7% per decade decline in cell counts of dopamine neurons in the nigra. Yeah, those studies are much smaller and they're tedious, they're hard studies. But let's accept that as a 4 to 7% rate per decade. We know they're declining. Why is this not actually greater? All evidence to date suggests that synthesis is actually increasing among the remaining dopamine neurons. As some dopamine neurons go bad, the neighboring neurons actually increase their synthesis capacity. This seems to work at least till the level gets too many neurons are gone. Once you get too many neurons gone, then it can no longer compensate enough. And a major hypothesis in Parkinson's is that you're seeing this decline well before you actually see symptom expression usually come up. And it's only after you've passed a certain threshold that those symptoms emerge. Again, in the aging realm, if you look in animal studies, the actual firing of dopamine neurons is also changing over time. Their spontaneous firing frequency is going down. More so in males than females, by the way. Males seem to lack some protective factor that females have. This shows up in Parkinson's too, where there's a substantially higher prevalence of Parkinson's in men than women. Now, what about Parkinson's and depression? There's a huge link here, in fact. Meta-analyses suggest that about 38% of patients with Parkinson's have depression. So it's way above the norm. Interestingly, among Parkinson's patients, women who have Parkinson's are more likely to get depressed than men who have Parkinson's, but the overall numbers of men who have Parkinson's so outnumbers the number of women that you see a lot of males with Parkinson's and depression, often with particularly high apathy symptoms being part of that picture. There's a little bit of data, which is interesting, in terms of linking Parkinson's pathology and depression. There are not a whole lot of studies, but they are consistent if you use DAT as your measure of dopamine system integrity presynaptically. Those with Parkinson's with depression tend to have lower DAT levels than Parkinson's patients who don't. And there are more studies which have looked at this correlationally, and essentially as those dopamine transporter levels are going down, the levels of depression are increasing. So now let me tie this all together here. I'm arguing from this that a declining dopaminergic tone that's associated with aging provides a vulnerability to specific aspects of depression. And those specific aspects of depression are those features of processes that we already know are well connected to dopamine function. I'm arguing that this is really something that's going to not be a general issue. This is not just, oh, dopamine causes depression. Rather, it's impacting how that depression is experienced. Now, you can come back to me and say, well, wait, shouldn't you're talking about this, but is there a dopamine deficit in depression in general? And I would argue there is not an evidence that suggests that depression is a dopaminergic disorder. If we look at meta-analyses of dopamine measures with PET, there's no effect overall that you see in terms of overall D2 binding. there's not an overall effect of dopamine synthesis other than that one paper that I noted about psychomotor retardation. In terms of that, if you look at the overall literature, there's not an effect. There does appear to be an effect with a small group of studies that use high affinity tracers. That's a much smaller population. I want to point out one particular study from that grouping, because one of those studies in that grouping of high affinities was with late life depression. And it's a study by Maria et al. It's a small study. I readily admit it's a small study, just 11 patients with depression versus 27 controls. They used a really great ligand, high affinity, called P2I. In the nucleus accumbens, they see a substantial reduction in the patients with late life depression. Now, I will note they did not do anything here to selectively recruit those patients based on symptoms. These are just individuals with late life depression, but they do see this effect, particularly in the nucleus accumbens, and then somewhat of an effect in the putamen as well. So a couple caveats. I've used the term dopamine tone, dopaminergic tone here. That's multi-determined. That's going to be a combination of dopamine synthesis. That's going to be impacted by receptors, transporters, firing properties. That could be multi-determined how those play out. You can think of this, though, in terms of dopamine synthesis. If those neurons are going down, we know you're losing those neurons. Synthesis is usually being compensated for. These are going to be cases where it seems to not be compensated for whatever reason potentially. You can think of this in terms of symptom networks as well. This could be either preferential symptoms. Yes, those are the symptoms like anhedonia, psychomotor retardation that are most affected. You could also think of this, though, in a network of symptoms. This could also be the starting point in some of these cases. That, for instance, the anhedonia is like the initial starting symptom, and then that starts to get expressed in terms of these other symptom domains. Because, in fact, if you look over here, loss of interest is a pretty central symptom within the network of symptoms. And this is actually specifically a network from late life depression studies. Couple other caveats here. If this is being driven really by dopamine declines, as I hypothesize, and men are more vulnerable to these declines than females, you might expect this to show a real change in prevalence of depression towards more males showing up in late life. And the data does not particularly show that change. That would be an argument potentially against my hypothesis here based on epidemiology. Readily acknowledge that. Second, I want to say, I can't emphasize it enough. I'm not proposing a model that says that depression is a dopaminergic disorder primarily. Depression is so heterogeneous. For instance, if you look at Eiko Fried's data on constellations of depression symptoms, he's like, there's so many different combinations of symptoms, and none of them, these are the 30 most common ones, none of these in his sample have a frequency of more than about 2%, because it's so complicated. The disorder is heterogeneous. And even if we talk about this in late life, you've got some other things that may particularly come up that may be causal. For instance, we talk of a vascular depression. And people have defined a vascular depression specifically in individuals over 50 who start to develop lots of white matter hyperintensities. And you particularly see this strongly, for instance, in those who have a lot of white matter hyperintensities in frontal cortex. That is, a unique domain, a unique route potentially into this. So I'm not claiming universal effect here. How much time do I have? I know probably not much. You were looking at your
Oh,
watch, Ken.
sorry, I got to, you have 9 minutes.
Oh, I'm perfect. I timed it excellent. How about that? Treatment implications. I think there's some really significant treatment implications here. One would be that maybe you want to do things to try to enhance dopaminergic tone in individuals with late life depression, particularly those showing psychomotor retardation and anhedonia. There are some studies that have particularly used methylphenidate as a way of targeting dopamine transporters to increase dopaminergic tone. Most of these have done it while adding this to SSRIs, so as a supplement rather than necessarily doing it alone. I will note reviews of this literature show you very clearly that a lot of these are small N studies. One of the largest is a study by Levertsky et al., where they had 143 depressed patients, double-blind randomized control study, really well executed. If you look at my lines here, I realize this is sort of hard to see, but this top line here is, this top line here is methylphenidate alone. This one is citalopram plus placebo. This bottom one, which is the square, is the combination where you've added methylphenidate to SSRI, essentially getting the largest effect. There was a recent meta-analysis by Juring where they took a whole bunch of studies. Some of these are decent quality, some of these are weaker quality, but overall one sees a modest effect, but consistently an improving effect, even if modest. Really essential to note, not a lot of randomized, this is just in general depression. This was not specific to late life depression. There's no targeting here across these studies specifically based on symptoms, which is what I believe you would really want to do in selecting the patients individually with a more precision medicine approach to target this. Some other studies have started to look at this using L-DOPA. There's actually some older studies that did this too. Really interesting study that unfortunately had to be retracted. And I almost never talk about a retracted study, but I can't resist it in this particular case. This was a 36 patient study with late life depression, who particularly this patients had lower gait speed. That was a requirement for entry in here. And they gave L-DOPA and improved depression as well as both gait and psychomotor speed. It was retracted because there were all sorts of protocol irregularities. There were issues related to their antidepressant washout, which wasn't entirely consistent. There were some issues they had in terms of inclusion, exclusion. They decided that it was enough so that rather than providing new analyses that they retracted the paper, which is unfortunate because I think the data itself was really quite interesting. We, influenced by that study, Warren Taylor has had a study we've been doing, which is specific to late life depression, which once again, I don't seem to be able to distinguish between LLD and LDD. We'll see what white matter hyperintensity is that I have. And clinically, with this study, the patients are selected because they have either anhedonia and or psychomotor slowing. We treat them with L-DOPA. They get PET imaging so we can look at what's happening within their dopaminergic system. I'd love to be able to report data on that. We're not there yet. Another year or so, but I'll be really curious to see how that plays out. I want to mention one other treatment implication, which is really in some ways less specific to a dopaminergic hypothesis, but we already know that SSRIs are less helpful in treating these sorts of reward processing deficits. Also, traditional CBT, as I mentioned earlier, Michelle Prask has argued is less effective. she's recently developed this really interesting new treatment called positive affect treatment, and has shown some good data validating it. And the idea here is to really try to enhance positive affect through psychoeducation, like positive affect labeling, followed by planning and practice of pleasurable activities, lots of imaginal recounting just about savoring pleasurable moments. They do a bunch of cognitive exercises about attending to positive aspects of experiences, self-attribution of positive outcomes, imagining future positive events, and then exercises to sort of cultivate and savor positivity through gratitude and generosity. This to me is something where it's like, well, if you've got core problems in late life depression with anhedonia, this is the sort of treatment that you should be thinking about augmenting. Okay, I've got just one other thing that I want to throw in here, which I wasn't initially, but I couldn't resist, given the context, which is just a study that my former postdoc, Lynn Dang, did when she was in the lab, where she came in one day and said, you know, there's this data that physical activity in patients with Parkinson's can actually alter D2 binding. And that you cannot, they've also shown this some with methamphetamine. It's like, what? You can do that? And so what she did was within our subjects, and we had a wide range of age in our subjects, is she basically did actigraphy with them. And she had them doing this for 10 days, and then she looked at correlations just between D2 binding and levels of activity. And those at higher activity actually showed higher levels of D2 binding. Now, there are a whole bunch of caveats I got to say with this. This was not a longitudinal study. We really, to say that this is having an effect, we need a longitudinal study to determine that. You'd also love to have more than just D2 receptors for that. You would love to have synthesis measures. You'd love to have DAT. Being able to do that longitudinally is tricky, just because NIH never has really understood that you need to fund longitudinal studies with 10-year grants, not 5-year grants. That said, I think this is a really cool finding and might actually eventually have some protective aspects to it. And anytime you can do something like that, which would also have cardiovascular benefits, to me, that's very, very intriguing. So just in conclusion, I've made an argument here that late life depression requires specific attention compared to other types of earlier life depression, and may have specific characteristics linked to dopamine that have treatment implications here. And with that, I will wrap up and just note many people are really essential to this work across multiple institutions, and thank both NIA and NIMH for funding this work. Thank you. Questions? Yeah.
So, if you look at people who developed depression in earlier life, but who go on to continue to suffer into later life, does their mix of symptoms change in the way that you might anticipate, given what you're telling us to do?
Yes. So the answer overall from the data in general says yes. However, I need to make one caveat there. Most of those studies do not actually separate in their analyses between those who had a first episode in later life and those who didn't. And so I've seen very few studies where they actually made a direct contrast that would, be really helpful.
Interesting contrast.
Yeah, I agree. Yeah, in back. So when you ask that question, you mean how would that actually play specifically into the dopaminergic side?
Yes.
Yeah, and so it's a great question. The answer I would first say is I don't know exactly. I mean, one possibility is that you're actually that this neuroinflammation component that some people are now studying could be a linkage in there. However, I actually see these as relatively distinct hypotheses when it comes right down to it, that the vascular is sort of one avenue here, and this dopaminergic component is a different avenue. And there may be some crosstalk there, but I actually see them as being different. I'll note in the molecular psychiatry paper, sort of in a little bit of deference to people like Alexa Poulos, we actually very early mentioned the vascular hypothesis as one of the things that's notable about wave-like depression. But then we veer straight off of that and never really come back and bring it back in because we don't actually have a clear linkage in terms
I don't have any citations on the top of my head right now, so I can't like prove it, but dopamine regulates a lot of the parts of the vascular system, actually. Yeah. I can give you citations, just not right now. Yeah. Yeah, both like centrally and peripherally. There's different, like the D1 does like one thing, and the D2, 3, the D5 handle like a different system. Like I said, I can see like the picture from the publication, but yeah, I can send it to you.
So could you disentangle whether the reduced expenditure of effort in depressed patient is due to lack of motivation toward specifically toward reward or fatigue in general?
It's a really good question because this is actually something we were always very concerned about within our paradigm of whether what we were picking up on was really a fatigue problem versus an effort discounting issue. The way we've tended to try to go about that is by doing things like entering in the trial number and into the analysis. And at least when we've added that into the computational model, the effort discounting aspect of it remains a driving component and not a fatigue component in there. Now, I've been particularly careful, though, in some of our paradigms to make sure things are set up in a way that they're not just having every, like they've got some little breaks. And it's fascinating, actually, on like our finger motions, how quickly we return to a sort of baseline default level and are ready to go again. And if you just think, when you're working at your computer typing, do you ever get to a point where you're like, my fingers are too tired to type? Usually not. Now, you're not only typing with your non-dominant pinky. I readily acknowledge that. But it's remarkable how quickly that sets. It's very different than like you're doing push-ups. Like, you do your sets of push-ups and, I don't know about you, okay, I can do my first 20. You ask me to like keep on, get next. It takes me a while before I'm ready to do that next set. The fingers, allow us some freedom there, I think, that makes that less likely of it to just be a fatigue. But when we've modeled it, we don't actually see really strong evidence that it's a fatigue effect rather than this effort, a willingness to expend effort. Now you can say, is that willingness to expend effort, is that different than motivation? And the argument is, well, it's directly being, helping compute subjective value. And the more your effort discounting, the lower that subjective value is, the lower you are motivated to pursue that. And so we really don't think you can explain this simply as a fatigue issue. But like we were really concerned, particularly in our older subjects that we're running now about this issue. And I initially did some revisions of the effort task that I did not pilot. And it was a mistake because I made the task actually too easy. And I had people just like every time they're just going for everything. And I had to recalibrate it again because our original had been all calibrated with younger adults. And so we had to play with that a little bit. What we often do, by the way, is the original task had just set numbers of what you had to do. We later realized that it was much better if we titrated it to people's actual response rates, what they could do, and set things as a percentage of each person's max response rate, rather than just saying, yeah, this is how many you have to do, which should have been obvious, but especially once we were starting to work with older adults, it's like, oh, well, duh, yeah, I can't ask someone to do something that they can barely do to begin with. I had to adjust that.
Are there any questions online?
There are no questions online.
Kate has a question though, and she's not online.
I'm not online. So I was gonna ask about this before you added in the slide, but I just want to ask it anyways. So you mentioned inflammation, you mentioned dopamine, you mentioned vasculature, you talk about executive function, I would say some of it related to the campus, and then you bring about aerobic exercise, right?
Yeah.
So really aerobic exercise does help dopamine, but it helps all of these other features.
Yeah, that's
right?
true. Yep.
So do you think that that is a very promising way forward to focus on maybe actual brain health writ large for individuals and sort of prevention or treatment or adjunct treatment of late life depression to address like sort of these multiple routes of mechanism by reducing inflammation, like potentially improving working memory, vasculature, et cetera, et cetera.
Yeah, and this may end up sounding like a cop out in a certain respect, but I always think things that generally are going to improve brain health are a really good idea across all sorts of different disorders and just healthy aging. I do think that we're looking at some things which may specifically be useful in or have some preferential benefit to some specific symptom areas and may have some preferential benefit to the dopamine system. But to me, it's like, hey, if you can help the dopamine system and help all these other domains as well, well, you should be thinking about that too. And whether you're talking about cardiovascular health or you're talking about something specific to a given neurotransmitter system, I think that all makes sense to be doing that. So yeah, I mean, if I wouldn't say you should only do aerobic exercise because it might do something healthy to your D2 receptors. There are a whole lot of other benefits there, without a doubt. I tend to, think from a like precision medicine standpoint of, well, here are the symptoms I'm really trying to target. But if I can get a whole bunch of other benefits out of it, absolutely.
All right. Let's have one more round of applause. Thank you.